Hello guys, let's go to the introduction. Thomas Hunt Morgan, an American genetic, is one of the most influential figures in the field of genetics. Thomas Hunt Morgan was awarded the Nobel Prize in Physiology or Medicine in 1933. Morgan's pivotal work in genetics involved using Drosophila melanogaster to understand heredity. His discoveries provide essential evidence for the chromosomal theory of inheritance, greatly advancing genetic science.
Description: Discovery of the white eye mutation, 1910. Morgan observed a male Drosophila melanogaster with white eyes instead of the common red eyes. This mutation provided a unique opportunity to study genetic inheritance. He crossed this white eye male with red eye females, producing all red eye offspring in the F1 generation. This indicates that the red eye trait was dominant. Next, F1 and F2 generations. Morgan then bred the red eye F1 generation flies with each other. In the F2 generation, he observed a 3:1 ratio of red eye to white eye flies. Crucially, all white eye flies were male. This led Morgan to hypothesize that the gene for eye color was located on the X chromosomes, with males XX expressing the white eye trait if they inherited the mutant genes of their single X chromosome. Third, reciprocal crosses to confirm hypothesis. Morgan conducted reciprocal crosses to test his hypothesis. Cross 1. White eye female XWXW with red eye males X plus Y produce red eye females X plus XW and white eye males XWY. Cross 2. Red eye females X plus XW with white eye males XWY produced a 1:1 ratio of red eye to white eye flies, confirming the X-linked inheritance pattern. Linkage and recombination. Morgan discovered that certain genes did not assort independently because they were located on the same chromosome, genetic linkage. He also found that linked genes could be separated during meiosis due to crossing over, where segments of homologous chromosome exchange pieces. His student, Alfred Sturtevant, used recombination frequencies to create the first genetic map, showing the relative position of genes on a chromosome. Confirmation of chromosome theory. In 1915, Morgan and his colleague published The Mechanism of Mendelian Heredity, detailing their finding and providing strong evidence of the chromosomal theory of inheritance. This theory posits that genes are located on chromosomes and are the carriers of genetic information. In 1904, Morgan joins Columbia University as a professor of experimental zoology. In 1908, begins working with Drosophila melanogaster. In 1910, discovery of the white eye male fly. Initial cross with red eye females shows red eye are dominant. 1911, F1 generation cross result in a 3:1 ratio of red eye to white eye flies in the F2 generation, with all white eye flies being male. Morgan hypothesized the X-linked inheritance pattern. In 1911 until 1913, conducts reciprocal crosses to confirm the X-linked inheritance pattern. 1913, discovers genetic linkage and crossing over. Alfred Sturtevant creates the first genetic map. 1915, published The Mechanism of Mendelian Heredity. 1933, Morgan is awarded the Nobel Prize in Psychology or Medicine for his discoveries concerning the role of chromosome in heredity.
Here is detailed methods for case study. The first case study is identification of sex-linked traits. It is about discovery of sex-linked inheritance pattern in Drosophila. The next case is genetic recombination and linkage map. It is about construction of the first genetic linkage map in Drosophila. There are 5 methods.
There are 2 types of articles. Firstly, genetic mapping in the age of geometry. Thomas H. Morgan, a biology revolution genetic thought his work on genetic mapping using fruit flies. His research supported the chromosome trophy of insect chain-chromosome relationship and laying the foundation for modern geometry. Awarded the Nobel Prize in 1933, Morgan continued to influence our understanding of the human genome and genetic base of their disease. Second article about fruit flies human head. It is about genetic recombination study purely by research like Thomas H. Morgan using flying have a play current role in understanding gene. It is about by unveil the message of genetic recombination, researcher has been able to identify disease caused the gene.
There are 5 methods that has been used by Morgan. The first one is selection of specimen. Morgan selected the fruit fly Drosophila melanogaster for his experiments due to its short life cycle, ease of breeding, and observable mutation. The next is initial observation. Morgan noticed a male fruit fly with white eyes instead of the normal red eye. This was a rare mutation, provided a starting point for his experiment. The third is crossbreeding experiment. Course 1. Morgan crossed the white-eyed male mutant with a normal red-eyed female. F1 generation result, all offspring, both male and female, had red eyes, indicating that the red eye trait was dominant. The 4 is F2 generation. Morgan then bred the F1 generation flies red eye among themselves. Observation in F2 generation, he observed a 3-to-1 ratio of red eye to white eye flies, but notably, all the white eye flies were male. The last method is data analysis. Morgan analyzed the result and hypothesized that the gene for eye color was located on the X chromosome. The conclusion is since males have only one X chromosome, XY, the presence of the white eye mutation on the X chromosome results in the white eye. Female XX with one normal X chromosome exhibited the dominant red eye trait. The result is this experiment provided clear evidence of sex-linked inheritance and established that specific genes and carry on specific chromosomes. In the diagram 1 is fruit fly, known as Drosophila melanogaster. The diagram 2 is sex-linked inheritance pattern in Drosophila. The first method is selections of multiple mutations. Morgan and his team selected fruit flies with multiple observable mutations such as eye color, wing shape, and body color. The second is breeding experiment. Initial cross flies with different combination of mutations of produce F1 progeny, while F1 generation crossed the F1 generation to produce the F2 generation, which would display a variety of combination of the original mutation. The third method is counting offspring. Morgan and his team meticulously count the number of offspring displaying each combination of traits. They note that certain combinations of traits appear together more frequently than others. The fourth is calculating recombination frequency. The frequency with which certain combinations of traits appear together recombinant types was used to calculate the genetic distance between genes. Recombination frequency formula is below this. Genes that were closer together had lower recombination frequency, while genes that were further apart had higher recombination frequency. The last method is constructing the linkage map. Using the recombination frequency, Morgan constructed a genetic linkage map showing the relative position of genes on the chromosome. The result is this methodology established that genes are arranged linearly on chromosome and that recombination frequency can be used to map their position. So here is genetic linkage map of the fruit fly Drosophila melanogaster.
How Thomas H. Morgan helped improve genetic embryology. By conducting statistical study of the way genetic traits are passed on in fruit flies, Thomas H. Morgan broke new ground in genetics during the first decade of the 20th century. His work confirmed that genes are stored in chromosomes inside cell nuclei. Next, confirmation of the chromosomal theory of inheritance. Thomas H. Morgan was able to provide a physical basis for heredity from his experiment by using fruit flies to observe the genes that reside on chromosomes. Next, Thomas H. Morgan was able to explain that some traits inherit differently between male and female by discovering cell-linked traits. Next, genetic mapping. Thomas H. Morgan, with the help of his student Alfred Stuyvesant, was able to create the concept of genetic linkage and genetic map. This discovery makes it possible to estimate the distance between genes on the same chromosome.
There are few applications that has been discovered by Thomas H. Morgan. The first one is genetic mapping and chromosome theory. Morgan's experiments with Drosophila led to the discovery of sex-linked inheritance and the concept of linked genes. This work supported the chromosome theory of inheritance, which posits that genes are located on chromosomes. Morgan's lab developed first genetic maps showing the arrangement of genes chromosomes, a foundational tool in genetics. The second, models organism development. Morgan's use of Drosophila established it as a major model organism in genetic research. The ease of breeding and observing mutation in fruit flies made them ideal for genetic studies. This legacy continues today as Drosophila remains a key organism in genetics and development biology research. The third application is educational impact and ethical standards. Morgan's mentorship and the collaborative environment he fostered in his lab influenced many prominent geneticists, including Alfred Stuyvesant and H.J. Muller. He emphasized open communication and the free exchange of ideas, setting ethical standards that persist in the scientific community. The last application is broader applications and influence. Morgan's discoveries laid the groundwork for understanding genetic linkage, recombination, and gene mapping, which are crucial in fields like medical genetics, evolutionary biology, and biotechnology. His work has had far-reaching implications, influencing the development of genetic engineering and genomics. There are some advantages that can be found due to application of Thomas H. Morgan. The first one is foundation for modern genetics. Morgan's work provided strong evidence for the chromosomal theory of inheritance, which states that genes are located on chromosomes and are transmitted during cell division and sexual reproduction. This laid the foundations for modern genetics. Next, development of model organisms. Morgan's experiments with fruit flies led to the discovery of sex-linked traits, where certain characteristics are determined by genes located on the sex chromosomes. This was a significant step forward in understanding patterns of inheritance. Next, innovative research techniques. Morgan's experiments allowed for the mapping of genes along chromosomes. By studying the frequency of crossover events during meiosis, he was able to determine the relative distances between genes on chromosome, establishing the concept of genetic linkage. Last, ethical standards in research. Morgan's investigations into mutations in fruit flies provided valuable insights into the mechanisms of genetic variability and evolution. Understanding how mutations arise and spread within populations is fundamental to comprehending the processes of adaptation and speciation.
Disadvantage number 1. Limited genetic models initially. Findings in fruit flies do not always translate directly to human due to species differences. This can limit the applicability of some genetic research conducted on Drosophila to human biology and medicine. Number 2. Simplification of genetic complexities. Morgan discoveries paved the way for genetic manipulation and engineering, which raise ethical concerns. Issues such as genetic privacy concern and the potential for eugenics are significant ethical dilemma that have arisen from the ability to manipulate genes. Number 3. Complexity of human genetic. Human genetic is far more complex than the simple Mendelian inheritance pattern observed in fruit flies. Polygenic traits, gene-environment interaction, and epigenetic add layers of complexity that were not addressed by Morgan's early work, highlighting the limitation of his models in explaining human genetic diseases. Number 4. Oversimplification of genetic concept. Early genetic models, including those developed by Morgan, sometimes oversimplify genetic concept. This can lead to misunderstanding about how traits are inherited and the influence of genetics on behavior and disease, potentially resulting in misconception and oversimplified conclusion.
For conclusion, in a nutshell, Thomas Hunt Morgan's groundbreaking work which revolutionized our understanding of genetic and heredity. His discovery about the role of chromosome in inheritance earned him the Nobel Prize in 1933 and have had a lasting impact on science. Morgan's research laid the foundation for modern genetics, and his legacy continues to influence and inspire research today.